Today we're going to be looking at a very exciting topic today, and it's still under reproduction in plants. And the topic today is development of new seeds. Under reproduction in plants, we have actually talked about quite a lot of, or discussed a lot of things about plants, looking at the structure of plants, looking at also the structure of a flower, which is actually the reproductive part of the flower of the plant. And we've also seen one or two things as it regards the different types of plants we have. We've also talked about the process that actually precedes fertilizations in flowering plants, and which we called pollination. But today we're actually going to be looking at another aspect of reproduction in plants, and that is development of new seeds. Development of new seeds. Now, in this particular topic, we will be actually exploring a whole lot of things or new issues. We will be looking at the structure of pollen grains. Remember when we were discussing looking at the structure of the flower, we said that the flower has what we call floral parts. And in these floral parts, we mentioned the sepals, we mentioned the petals, but we also mentioned some vital aspects of the flower, which has to do with the stamens and the carpels. Another name for the stamens, we said, is called the androecium. And then another name for the carpels or the pistil, we called it the gynosium. Okay, and I told you that the stamens actually consist of an anther and also a filament. Now, the filament is actually a slender tube that holds the anther, and then inside of the anther is the male sex cell or male gametes of flowering plants, and that is the pollen grains. So today we're going to be looking at the structure of the pollen grains. We're also going to discuss one or two things about the pollen grains. And then also number two, we will be looking at the structure of the ovules. Also, let me take your mind back to what we discussed earlier in our previous videos. We said that the ovules is actually located in the ovary of flowering plants. And the ovary is actually a part of the carpels. The carpels, also known as the gynosium, or which you can call the female reproductive part of a flowering plant, it actually consists of the stigma, which is on top. It consists of a style, which is a tube that links the stigma to the ovary. And inside of the ovary, we have the ovules, which contains ovum or eggs that must be fertilized in order to give rise to young individuals or young plants. So we're going to be looking at the structure of the ovules. We are also going to see the process of fertilization in flowering plants. Now, how fertilization takes place in flowering plants. If you compare that to reproduction in animals, we also looked at the process of fertilization in animals. We also talked about pregnancy, childbirth, and all the others. If you go to some of our videos, you're going to see that. So, but this time around, we're going to be looking at fertilization in flowering plants. And then we're also going to be talking about development of embryo, development of the embryo. Now, after the fertilization, or after fertilization has actually taken place, zygote is formed. Now, what happens to this embryo? What are the processes of development? What makes up the embryo? And then we're also going to be talking about germination of seeds. We're going to be looking at the types of germination. We're also going to be looking at the conditions that are actually necessary for germination. And then we're going to see or note what it means for a seed to be dormant. That is seed dormancy, and how to actually break seed dormancy. So these are the things or subtopics we're going to be looking at in this topic, development of new seeds. Now, as our culture, our specific objective has to do with things you should be able to present, or things you should be able to, let me say, explain or do activities you should be able to come with by the end of this particular topic. So by the end of this topic, you should be able to draw the structure of pollen grains and the ovules. Now, the reason for the drawing or the diagram is so that you can be able to identify parts of the pollen grains as well as the ovules. Number two, you should be able to describe the process of fertilization. It is actually very important. Most questions you see scattered in WAEC and also in JAM, they ask questions on some of the hidden things or hidden explanations in the process of fertilization in flowering plants. And also number three, you should be able to describe a developing embryo in flowering plants, what actually happens, what actually these embryos are made up of. And then also you should be able to define what we call germination of seeds and also mention the types of germination. And then also you should be able to mention some of the conditions that are necessary for germination to actually take place. And then finally, you should be able to define what we call seed dormancy and also mention the ways seed dormancy can be broken. Seed dormancy can be broken. So these are actually the areas we're going to be, I would want you to be able to tell or give answers to by the time we end in this particular class. And if you're actually ready, I'm very set. So let's move to the first thing we're going to be looking at, which is development of new seeds. Development of new seeds. Remember, we said something about in reproduction in plants, in animals. We said that for sexual reproduction to actually take place, there must be a production of what we call gametes or sex cells. In terms of humans, or mostly invertebrates, we said the sex cells include the sperm cell, which is from the male, and then we have the egg cell, which is from the female. Also in flowering plants, since they undergo sexual reproduction, flowering plants also have their sex cells or gametes. They have a male gamete and there is also a female gamete. The pollen grains, which is located in the anther of a flower, is the male sex cell of flowering plants, or you can call it the male gamete of flowering plants. While the ovules is the female sex cell of flowering plants, or you can say it's the female gamete of flowering plants. And it is these pollen grains and ovules that are actually involved in the formation of the zygote, or of a zygote in flowering plants. Now, it is also very important to take note of this. Remember, we also said in reproduction in mostly humans, we used humans as our case study. We said that the gametes, they are all haploid cells. Why? Because during the formation of gametes, these sex cells undergo what we call meiotic cell division. In the same manner, in flowering plants, when these sex cells are being produced, the sex cells undergo meiotic cell division. It simply means that the pollen grains, if you check their sex cells, they are also haploid cells, while the ovules are also haploid cells. And when a haploid cell combines with another haploid cell to give rise to a zygote with a diploid cell. Okay, so please take note that the sex cells or the gametes in flowering plants, they are also haploid cells. All right, now the next thing I want us to look at is the structure of the pollen grains. Remember, I said the reason for us studying the structure of the pollen grains is so that you can be able to identify most of the things that make up the pollen grains, because they play a major role in the process of fertilization. Now we have look at the structure. Now the two things I want to actually point out in this is the exine, which is the outer membrane that protects the inner structure of the pollen grains. the intine, which is a dense cytoplasm, and the nucleus, which plays the major role in fertilization. It plays a major role in fertilization. So that is actually the structure of the pollen grains. So in other words, we can say that the pollen grains actually consist of three major parts. It consists of an outer membrane, which is the exine. It consists of a dense cytoplasm or matrix, which is called the intine. And then it consists of a centrally placed nucleus. Centrally placed nucleus. Of course, you know that the nucleus contains chromosomes, and these chromosomes, they contain genes, which contains hereditary materials that can be passed from parents to their offsprings. All right. Now, the next one is the structure of an ovule. Remember, we said that the ovule is the female sex cell of flowering plants. So that is the major structure. That is the structure of the ovule. That's the structure of the ovule. Now, apart from all the things we can see here, there are several things I want us to actually take note of. Now, one of them is take note of where the ovules is actually located. Now, there is an opening, if you could see that, there is actually an opening which is called the micropyle. It's a tiny opening that leads into the ovum. Now, the whole structure here is the ovary, all right? Or you can call it the ovule. That's the ovule. And within the ovule, we have the egg cell, which is responsible for fertilization. And inside of it, you can see the definite nucleus, which is the secondary nucleus located here. And all these play major roles during fertilization processes. Now, an ovule consists of the hilum. Now, the hilum is the point of attachment to the placenta. The point of attachment to the placenta. The ovule also consists of the integument. The integument is actually a covering over the ovule. It has an embryonic sac, it has a nucellus, it also has a micropyle. And I showed you that the micropyle is actually a tiny opening that leads into the egg cell. All right. Now let's move from the structures, since we have now noticed the structures of these sex cells, both the male sex cell and the female sex cell in flowering plants. Let's proceed to the process of fertilization in flowering plants. Now, before fertilization takes place in flowering plants, pollination must have taken place. Before fertilization will take place, pollination should have taken place. And now, what is actually pollination, if I may repeat again? Pollination is the transfer of pollen grains from the anther to the stigma, or transfer of mature pollen grains, yes, from the anther to the mature stigma of a flower, of another flower of the same plant, of another plant, but of the same species. So this is what we call pollination. So until the pollen grains lands, or a mature pollen grains lands on a mature and suitable stigma, pollination has not yet taken place. So if pollination is successful, after pollination, the pollen grains that has landed on a mature stigma absorbs sugary liquids on the stigma. So on the stigma, there are sugary liquids. So once these pollen grains land on the mature stigma, it begins to absorb these sugary liquids. And as it absorbs it, it begins to swell and also germinate. It begins to swell and also germinate. Now, while it is undergoing this process of germination, certain things continues to take place inside of the pollen grains. The nucleus inside of the pollen grains divides into two nuclei. It divides into two nuclei. Now, these two nuclei are the large tube nuclei and the small generative nucleus. The large tube nucleus, rather, and the small generative nucleus. Now, as the pollen grains also undergoes this swelling and germination because of the absorption of the sugary liquid, what happens is that the exine, which is the outer membrane of the pollen grains, splits. Once it splits open, pollen tubes begin to grow out and down. So it grows out from the pollen grains, and it grows down through the style. Through the style, you can see that on the screen. Through the style, it passes through the style, and as it goes down through the style, it passes into the ovules through the micropyle. Remember, I told you that the micropyle is actually a tiny opening, which of course allows pollen tube to enter into the ovules. So once this happens, this pollen tube passes through the micropyle and then gets into the ovules. Now, the generative nucleus, remember, as this happens, the generative nucleus also follows this pollen tube and continues through this tunnel or channel and enters into the ovules. But before it gets to the ovules, the generative nucleus further divides into two male nuclei. Now, these two male nuclei are the ones that enters into the ovule and enters inside the embryo sac. Now, one of the male nuclei fuses with the egg, which is the ovum, to form a zygote. Now, this is what we call the first fertilization. One of the male nuclei enters into the ovum and fuses with the nucleus to form the first, or to form a zygote, and this is what we call the first fertilization. Now, while the second male nuclei enters into the embryo sac also and fuses with the secondary nucleus. It fuses with the secondary nucleus to form what we call an endosperm. This also is the second fertilization. Please take note, in flowering plants we have double fertilization. So sometimes you might be asked to explain double fertilization in flowering plants. Now, how did this double fertilization once again come about? Is from the generative nucleus. Once they pass into the pollen tubes, I said the generative nucleus splits into two male nuclei. One of the nuclei enters into the ovules and fuses with one of the nucleus of the ovum, of the embryo, to form a zygote. All right, and this zygote definitely develops into an embryo. So that's the first fertilization. The second fertilization is that the second male nuclei fuses with the secondary nucleus. Now, once fusion is successful with the secondary nucleus, it forms what we call an endosperm. So this is the second fertilization. Hence, we say flowering plants undergo double fertilization. So this is what we call fertilization process in flowering plants. Now, once a zygote is actually formed, the zygote undergoes further development by mitotic cell division to form what? An embryo. Remember, development and growth, cell division must be involved, and the cell division that is involved during growth and development is mostly mitosis. But the one that is involved during formation of a gamete is meiosis. Okay, so the zygote develops and grows into an embryo. Now, by mitotic cell division. Now, this embryo consists of what we call a plumule. It consists of a radicle. It consists of one or two cotyledons. Cotyledons are simply called seed leaves. And sometimes it contains an endosperm. If it is one cotyledon, you will see an endosperm. But if the embryo has two cotyledons, you won't see an endosperm. Please take note of that. Now, the plumule can also be called the embryonic shoot, or it is what forms the stem. Okay, once the embryo starts developing into a young plant, it is the plumule that develops into a stem. So we call the plumule embryonic shoot. That is the shoot of the plant. While the radical forms what we call the roots. And so we call it the embryonic roots. And then the cotyledon, they form the leaves. That's why we call them the seed leaf. And that's what makes up a plant. That's what makes up a plant. While the endosperm, if it is a one cotyledon, there is presence of an endosperm. And the endosperm is used by the embryo for its nourishment and growth. Okay? So please take note. After fertilization, the ovules develop into a seed, develops and becomes a seed. Please listen. After fertilization, the ovules develop and becomes a seed. But after fertilization, the ovary develops and becomes a fruit. So the ovary, which contains the ovule, becomes the fruit, while the ovule inside of the ovary after fertilization becomes the seed. And then the embryo is found inside the seed. So you have a fruit which carries a seed, and inside of the seed you have an embryo that is a growing or a developing young plant which has life. Okay, so once you get any particular fruit, note that in that particular fruit, if there is a seed in it, because there are, yes, if a particular fruit that has a seed in it also has an embryo. And that's why when you plant seeds on suitable soil, and it will definitely germinate and grow into a living organism or into a young plant. Why? The seed contains an embryo. The seed contains an embryo. So this is actually the process of development of the embryo. Now, let's go back to the seeds. We will be talking much about the fruits in subsequent videos, but let's concentrate on the seed. Now, remember we said that a seed can be gotten from a developed ovule, okay, after fertilization. So by definition, we can say a seed is a ripened, fertilized, and developed ovule. A seed is a ripened, or you can say a mature, fertilized, and developed ovule. A mature or ripened, fertilized, and developed what? Ovule. Now, a seed, remember I told you that what is found in a seed, embryo is found in a seed. But a seed doesn't only contain an embryo. Now, what makes up a seed? What makes up a seed? Now, a seed consists of what we call a seed coat. Another name for seed coat is called tester. So a seed consists of a seed coat or a tester. Now, the tester, it plays also a major role during germination of seed. We're going to be looking at the structure of a seed, but please pay attention to this. Now, a seed coat actually is a thin and transparent membrane that covers the embryo. A membrane that actually covers the embryo. Now, the next thing you find in a seed is the hilum. H-I-L-U-M. The hilum. Now, the hilum is a scar. Now, that scar is actually the point of attachment of the seed to the stalk. The point of attachment of the seed to the stalk or to the seed stalk. Now, the next thing you find also in a seed is the micropyle. Remember, I told you that a micropyle is a tiny opening or a hole, a tiny opening. Now, one of the major functions of the micropyle in a seed is to allow air and water to enter into the seed. Now, this air and water that enters into the seed are used by the embryo for their growth and development. And then finally, what you see in a seed is also the embryo. And this is the part that actually develops into a new plant. So a seed consists of a seed coat, it consists of a helium, it consists of a micropyle, and then it also consists of an embryo. These are what makes up a seed. Now, as you can see in the structure, we have a bean seed, and then we also have a corn seed. Now, not everything was actually displayed here, but look at this. We have the seed coat, which is the outer part of the seed, that it is like a membrane that covers the inner structure of the seed. Then we have the endosperm. Remember, we said if it is a one seed leaf or a one cotyledon, it has an endosperm. An example of a seed with one cotyledon is the maize seed or the corn seed. The one that has, example of the one that has two seed leaf is the bean seed. So bean seed don't have endosperms, but maize seed or corn seeds, they have endosperms. Then also we have the cotyledon, as you can see on the structure there, and then we have the embryonic stem and the embryonic roots that makes up the embryos. All right, so that's where the embryo is actually located. So this is the structure of a seed or a viable seed. Now, the next thing to take a look at, before the seed actually develops into a new plant, or the process, let me put it this way, the process by which this seed actually develops into a new plant or a young plant, we call that process germination. So what exactly is germination of seed? Now, germination of seed can be defined as the gradual change or development of an embryo into a young plant. Simple. The gradual development or changes in an embryo into a young plant. So that is what we call germination of seeds. Now, we have different types of germination of seeds. Different types. Number one example, or we have two types actually, and these two major types of germination includes, number one, we have what we call the hypogeal germination, and number two, we have what we call the epigeal germination. Hypogeal germination and epigeal germination. Now, what is hypogeal germination? Let's take a look at hypogeal germination. Now, hypogeal germination is actually a type of germination in which the cotyledon remains below the soil surface. The cotyledon remains below the soil surface. So that is what happens. It remains below the soil surface. Now, we will be briefly talking about hypogeal germination process, how it happens. Now, number two, or examples of plants that undergo, or seeds that undergoes hypogeal germination, they are all monocotyledonous plants. Monocotyledonous plants undergo hypogeal germination. Monocotyledon means one seed leaf. So plants with one seed leaf undergo hypogeal germination. Examples, we have maize, we have rice, we have wheat, we have millet. In fact, all the cereals and the wheats or the grains, they all fall under hypogeal germination. Okay, they fall under hypogeal germination. Now, what is actually the process of germination in hypogeal germination? Now, how does it happen? Now, when a seed falls in a suitable soil, what happens is that the seed absorbs water from the soil, and that water it absorbs from the soil causes the seed to swell. As it swells, the testa is splitted, or this water that the seed absorbs and swells splits the testa. Once the testa is split open, what happens is that the embryo inside of the seeds starts bringing out first its radicle. Now, the radicle starts going downwards. Always the radicle grows downwards. Always the radicle grows downwards. Now, once this happens, the plumule starts also sprouting out from the seed, but the plumule actually grows upward above the soil. Now, if you look at the structure here, you can see in the first stage, the radicle going down, the plumule coming up, and you can see the plumule is out, and then the radicle is still out, but the seed itself still remains beneath or still remains inside of the soil. So this is the process of hypogeal germination. Now we have the next type of germination, which is called epigeal germination. Now in epigeal germination, there is a type of germination in which the cotyledon is carried and remains above. The cotyledon is above, that's the seed itself, is carried above the soil, and it remains above the soil surface. Okay, examples of plants that actually undergo epigeal germination, we have the dicotyledonous plants. And dicotyledonous plants simply means they have two seed leaves. Now examples include the beans, they have the cowpea, we have the groundnuts, we have mango, we have melon, and so on. These are examples of plants that undergo epigeal germination. Now, during epigeal germination, the process is as the same with the hypogeal germination. But the major difference between epigeal and hypogeal germination is that when the seed absorbs water from the soil and the testa or seed coat splits, the radicle goes downwards as usual, the plumule it goes upwards as usual, but this time the cotyledon, which is the seed leaf, is carried above the soil surface. Is carried above the soil surface, as you can see here. You see the radicle is produced going down, and then from the radicle you can see that the seed leaf is actually emerging out of the soil, and then it comes out of the soil surface and sprouts to release the foliage leaves, and this is what we call epigeal germination. This is epigeal germination, and like I said before, it is common in most legumes, mostly common in legumes, while hypogeal germination is mostly common in cereals. All right, now the next thing we're looking at is differences between hypogeal germination and epigeal germination. Very important question that usually pops up, theory questions that actually comes out. We have number one is that the cotyledon, in terms of hypogeal germination, we've just mentioned it, remains below the soil, while in epigeal germination, the cotyledon is always carried above the soil. And then another difference between hypogeal and epigeal germination is that the hypocotyle in hypogeal germination elongates, while the epicotyle in epigeal germination elongates. Another thing again we notice as a difference is that the energy obtained from the food is stored in the endosperm. Remember I told you in hypogeal germination, mostly monocotyledonous plants, which we give an example, the corn seed, they have the endosperm, and these endosperm are used by the embryo as food to obtain what? Energy. But in epigeal germination, the energy that is being obtained from the food is stored in the cotyledon and not in the endosperm, because there is an absence of endosperm in epigeal plants or dicotyledonous plants. Okay, so these are three main differences between epigeal and hypogeal. Now, moving ahead is conditions that are actually necessary for germination to occur. Now, there are several conditions. If these conditions are not taking place, that particular seed is likely to die. Now, not all seeds that are planted actually make it into a growing or grows into a young plant. There are several factors that actually contributes to this event or these happenings. Now, one of them is water or moisture. Now, if a plant does not get adequate water or moisture to activate the cells of the seed and help soften the testa, there's going to be a big problem. So the water that was being absorbed by the seed when it is inside of the soil, helps to activate the soil or the seed, and also helps to soften what? The testa, so that it can split. So water or moisture is very, very necessary. Another one is suitable temperature. Suitable temperature is also required for germination to actually occur. Another is air. Now, air is needed, which is the oxygen, is needed so that the embryo can carry out respiration. And one of the major purpose for respiration is to release energy for the growth of this embryo. Because what the embryo will do, as in order to develop into a seed, requires energy. Develop into a new plant requires energy. The production of the plumule, the production of the radicle, and all these, they require energy. And this energy can be gotten from both the food and also oxygen, because when oxygen acts on this food, they release what? Energy. Other aspects of other conditions or factors that are necessary for germination to take place, we have enzymes. Now, these enzymes are required in respiration, and also to speed up the rate of reactions within the cells of the seeds. And of course, another condition is food. The embryo actually needs food. Where energy are being stored. Now, food are stored in the cotyledon, mostly, like I said, in dicotyledonous plants or seeds, but in monocotyledonous seeds, the food, which the embryo obtains its energy, is stored in the endosperm. And then finally, one of the major things a plant or a seed is supposed to, one of the conditions that aids its germination is that the seed must be viable. The truth is, all these other conditions can be in place. If the seed is not a viable seed or a lively or active seed, that seed cannot germinate. No damaged seeds actually germinate, even if the food is available. Of course, if it is damaged, food might not be available. Enzymes might also be damaged if the seeds are also damaged. And so many other things can actually happen to a damaged seed. But please note, a seed must be viable before it can undergo the process of germination. Now, finally, before we go, let's take a look at what we call seed dormancy. Now, as the name implies, dormancy talks about inactivity. When something is dormant, it means it is inactive. So seed dormancy is the inactive period of a seed during which growth slows down or completely stops. So it is a period. It is a period in every seed. It is something that happens to every seed. But how quick this thing happens, okay? The length of this dormancy determines whether this seed can actually germinate or not. So seed dormancy is the inactive period of a seed during which the seed grows slowly or completely stops. Now, this is due to certain internal and external factors, which we will not be looking at today. But let's see how we can break seed dormancy. What are some of the ways through which seed dormancy can be broken? So if a seed is actually dormant, these are the things you do. Number one, remove the testa or the seed coat. Remove the testa or the seed coat. Another thing again is you expose it to fire. So it's not that you dump it in fire, you bring it close, not too much so as not to make it to kill it, but expose it a little to fire. Another one again is exposure to alternating high or low temperature. So make it a little bit high, low. So you alternate it. Another way also you can break seed dormancy is addition of dormancy breaking hormones. So there are some hormones that break seed dormancy. I think one of them is the gibberellins. We also have, I think, the auxins and the cytokinins. They also help to break seed dormancy. And then also we have treatment of seeds with acid or digestive enzymes or water. So you can use these ways to actually break dormancy in seeds. So this is where we call it a wrap up on this particular class. But definitely before we go, let's take a look at few questions using our Examguide app. But this time we're looking at UTME. All right. Now let's look at just a few questions. Look at this. They said a seedling grown in the dark is likely to be what? Is it likely to be stunted? Is it likely to be dormant? Is it likely to be sturdy? Is it likely to be etiolated? Okay, so the correct answer is it is likely to be stunted. So light is needed. Light is actually needed for it to grow. Respiration and so many other things is needed when light is present. Next one is number 2. We have, look at that structure. I think that gives us a very clear description. This is epigeal germination. If you go back to one of the things, things we posted on the screen concerning epigeal germination, you can see that the cotyledon is going above the soil surface. And you can see what is. So this type of germination illustrated here is epigeal germination and not hypogeal germination. And then number 3, water is necessary for germinating seeds because, remember we've just mentioned it, is it because it activates the enzyme? Is it because it wets the soil for proper germination, protects the seed from dissociation, promotes aerobic respiration? Now, water is actually necessary to activate the cell or the enzymes. It activates the cells and also enzymes. And then number 4, it says in a bean seed, absorption of water at the beginning of germination is through what? Now, it is actually through the micropyle. Remember, I told you that the micropyle is a tiny hole or opening that allows or permits the entrance of water and air or oxygen. So it passes through the micropyle. And then number 5, you can still see the same structure. They said the main function of the structure labeled 1. The main function of the structure labeled 1 is to pull the young shoot above the ground. Is it store food for the young plant? Is it store water for the seedling? Is it protect the young plumo? Okay, let's look at it again. A, is it to pull the young shoot above the ground? So that, if you check it, that is the cotyledon. And is it to store food for the young plant? That is the correct answer. So the cotyledon stores food for the young plant. If you go back to what we said about food, remember we said in the dicotyledonous plant, that is a dicotyledonous plant, beans, we said the food is stored in the cotyledon. But in monocotyledonous plants, the food is stored in the endosperm. Okay, it's stored in the endosperm. So this is the correct answer. And then finally, number 6, they said epigeal germination of a seed is characterized by what? Epigeal germination of a seed is characterized by, is it more rapid elongations in the epicotyl than the hypocotyl? Is it equal growth rate of both? Is it more rapid elongation of the hypocotyl than the epicotyl? The correct answer is more rapid elongation of the epicotyl than the hypocotyl. So this is where we call it a wrap up on this particular class on development of new seed. I'm going to see you in our next videos. Bye for now.